Now, second point is chromosomal aberration. Chromosomal aberration means what? If the structure of chromosome is disturbed or altered, then it is called chromosomal aberration. We can say change in the structure of chromosomes. Second, chromosomal aberration. That means what? Change in the structure of chromosome. Right? Many types are there. First one is our deletion. Deletion is also two types. On the basis of the site of the deletion, right? First, terminal deletion, we can say terminal deletion, right? Terminal means what? At the end, some part is deleted. And second one is intercalary deletion. Intercalary deletion. Terminal deletion and intercalary deletion. Example is given here. Suppose that here, these are chromosome. That one is chromosome. I write here with some words, as for example 1, 2, 3, 4, 5, right? Here also 6, 7, 8, 9. Okay, then what happens? Terminal part is deleted. Suppose that this part is deleted. Then what we have the new chromosome, it is only 3, 4, 5 and 6, 7, 8, 9. So it is deleted. This part is deleted. 3, 4, 5. That means called terminal deletion. Terminal means this part is deleted at one side. Do you understand? As for example here, cried, due, chat, CHAT, cried, due, chat syndrome. Cried, due, chat syndrome due to the terminal deletion in 5th chromosome. 5th chromosome. Cried, due, chat. Right? Second one is our intercalary. This one is first. And second one is our intercalary. Intercalary means if the deletion occurs not terminal side but central position in between, right? As for example here, same chromosome I write here 1, 2, 3, 4, 5, 6, 7, 8, 9. Then what happens here? It is not the first terminal part, not 1, 2 or 8, 9, but in middle. Middle means what? Suppose that this part is deleted. Right? Now new chromosome, it becomes 1, 2. Right? Then 6, 7, 8, 9. Look here, this part is deleted. This part is deleted. Right? That this is the new, is called as intercalary deletion. Intercalary deletion. Do you understand? Right? Intercalary means between two chromosomes, but it is not from terminal end but in between. Right? So this is the example of deletion. Then second one is our inversion. Inversion means what? When exchange occurs. Not delete is there, but they are exchanged. Exchange means they change their position, they change their site, right? Look here. Second is inversion. Inversion means when the position is exchanged, when position of gene is exchanged. It is also two types, same as to there, right? There we can say terminal or intercalary. Here another term is used, paracentric and pericentric, right? Two types. First one is our paracentric and second one is our pericentric. Paracentric and pericentric, right? Para means which is not present. Look here, we have two chromosomes. Suppose that this one is chromosome, another. Okay, I write this like this one. Then paracentric means what happens here? 1, 2, 3, 4, 5, 6, 7, 8. Suppose that change some I write here. This position is changed. 2, 3. Either one side or two side, that is not a matter, right? It can change. Now new chromosome, it becomes, no part is deleted here, but this position is changed. 1, 3, 2, 4. Look here, this number is changed here. 5, 6, 7, 8. So this one is called paracentric, right? And in pericentric, what happens when position is changed? Means these genes transfer to another arms, right? Here what happens? They are changed in, look here, this one is a chromosome. 1, 2, 3, 4, 5, 6, 7, 8. Now what happens here? Here the position of this 3, 4 and 5, 6, these is changed. Change means exchange between two different arms. Now what happens here? The new formed chromosome is 1, 2, 5, 6, 3, 4, 7, 8. Look here, these words are changed. This and this. Exchange. This one is called pericentric, right? Third is duplication. Duplication means understand repetition. Means in one chromosome what happens? The same number is repeated many times. That is called duplication, right? As for example, in where bar I character, in Drosophila, in fruit fly, scientific name is Drosophila, right? This one is present. Here what happens? Here, suppose that I write here, these are two chromosomes, and I write here 1, 2, 3, 4, 5, 6, 7, 8, 9. One chromosome is there, right? Second chromosome is also here. These are homologous chromosomes. Not these happens in within a single chromosome, but this happens within two different chromosomes, homologous chromosomes, right? At the time of when crossing over occurs, that time what happens? Some genes what happens? From one chromosome it is deleted and it add into the another chromosomes, right? When it add into the another chromosome, right? Another homologous chromosome. So what happens? This another homologous chromosome, it contains that is more than one, right? We can say it is a repeated DNA sequence. As for example, you can see here 1, 2, 3, 4, 5, 6, 7, 8, 9. Another chromosome is also this one. These are homologous chromosomes 1, 2, 3, 4, 5, 6, 7, 8, 9. At the time of crossing over in Pachytene condition, what happens? A new chromosome is formed. When they exchange their genetic material, what happens here? We can write here 1, 2, 3, 4, 5. First arm, no, second arm, it is also only 6, 7. Because 8, 9, it is transferred to the next one, right? This one in crossing over. The next chromosome, it contains 1, 2, 3, 4, 5, 6, 7, 6, 7, 8, 9. Look here, these 6, 7, 6, 7, they are repeated. So this one is called duplication. This one is called duplication, right? So this bar I character is present in fruit fly means Drosophila due to this duplication, right? Bar I character means what? This eyes becomes narrower than the normal eyes, right? Now after duplication, the third last one is our translocation. The third and the last chromosomal aberration. Translocation. Translocation means, you understand, translocation means what? Transfer of this. This translocation, one thing is there. It occurs between non-homologous chromosomes. That is important. It occurs between non-homologous chromosome. That means this one is illegal. We can say here, this one is the illegal mutation, right? Duplication means between homologous chromosome. Deletion or inversion means what? Between single chromosome, right? So this third one is our translocation. Third type. Not third, it is fourth type, I think, because deletion, inversion, duplication and translocation. Translocation is also called as illegitimate mutation, right? Illegitimate. Mate, or is called as illegal, right? Mutation. It is three types. Three types. Which three types are there? Translocation. One more thing. It occurs between non-homologous chromosomes. Between non-homologous chromosomes. That's why it is called as illegitimate or illegal chromosomes, right? This one illegal, illegal, right? Okay. So first one is our first is simple translocation. Simple translocation. Right? Second, interstitial or shift translocation. That is B. Interstitial, or we can say shift translocation. And third, it is, that is C. You can write here C. This one is reciprocal translocation. Simple translocation means what? Here, some part of one chromosome is deleted and come to the different part. Same as to duplication, but duplication occurs in the same duplication occurs between the homologous chromosome, but this translocation, it occurs between the non-homologous chromosome. Same process there. As for example, here I draw here. That is our first chromosome, 1, 2, 3, 4, 5, 6, 7, 8, right? And second chromosome, because they are non-homologous chromosomes, so they have genetic material is also different. I write here A, B, C, D, E, F, G, H, right? Then after what happens? After exchange of this one, suppose that this part is exchanged. Then what happens here? The first is it becomes 1, 2, 3, 4, and second part is coming only 5, 6 is there. But the second one, this one, it becomes A, B, C, D, and what happens? E, F, G, H, 5, 6. This. So this is the example of simple translocation, same as to duplication, but it is not duplicate because it is not repeated, because it contains different genetic materials, right? So that is it. This exchange occurs between the two non-homologous chromosome, so it is called as simple translocation, right? Second one is our interstitial or shift translocation. Interstitial means it is not at the terminal side, but it is in between. Same is there, but what happens? It occurs at the terminal side, this last one, but it occurs in between. That is called. That's why it is called interstitial, right? You can see here same thing is there. 1, 2, 3, 4. This side 5, 6, 7, 8. Another chromosome is this one. A, B, C, D, E, F, G, H. Then what happens here? When they exchange, the same thing is there. A, 1, sorry, this one is 1. 1, 2, 3, 4, 5, 6, right? And here what happens? A, B, C, D, E, F, 5, 6, G, H. You can see here, this one 5, 6 is present not at the terminal side, but it is present in between the segment. In between the segment means interstitial. Interstitial means between, right? This word interstitial. Okay, now this reciprocal, it is important. Reciprocal means what? Exchange. In exchange, what happens? Means they exchange. That means here what happens? Not here. Here you can say this one is an example of deletion. This part, it is this one. They shows this one and this one. They are the example of deletion also. We can say when we consider only one chromosome, so that is we can say it is deleted. But deletion is there, and here addition is there. We can say yes. We can say right. This is also deletion and addition is there. But in reciprocal, what happens? It is like to the inversion, right? This exchange of genetic materials. What happens here? Suppose that this one is our one chromosome. It is 1, 2, 3, 4, 5, 6, 7, 8, and this one is another chromosome, right? A, B, C, D, E, F, G, H. So after mutation, what happens? What happens here? It becomes 1, 2, 3, 4, and another side it is 5, 6, G, H. This GH comes from that side, and another chromosome it contains A, B, C, D, and this side it contains E, F. Now 5, 6, 7, 8. These are exchanged, right? So this one is called as this one. It becomes what? It is the example of reciprocal. These are changed. Sometimes this 7, 8 comes to that side, GH come to that side, maybe CD come to that side, right? So this one is called as, this one is example of reciprocal translation. And as for example for reciprocal translation, it is CML, we can say chorionic myeloid leukemia. This one, here I write leukemia, CML. It is an example of this reciprocal translocation. Here what happens? That exchange between 22 and that is second one is 9th, right? 22 and 9 chromosome. Here what happens? Exchange, EXCHANG, exchange between 22 and 9th chromosome. You understand? So these are the different type of chromosomal mutation. Now here gene mutation. Okay. The gene mutation is not so much. Gene mutation is given in our NCERT already. You reviewed this one. Once more here we are going to gene mutation. Like to this chromosomal mutation, gene mutation is also two types, right? So two types means what? We can say here substitution and frame shift mutation. Substitute and frame shift mutation. As for the name is given, substitute when it is replaced or when it is exchanged with purine or pyrimidine nitrogen base, right? And frame shift mutation with what? When the total frame shift, when the total number of nucleotides are changed, right? We will read here. Now this is the second gene mutation. Gene mutation, two types. Which two types? First one is substitution and second one is frame shift mutation. Substitution and frame shift mutation. Now this substitution means replacement of one nitrogen base. You can see here replacement of one nitrogen base, right, with another nitrogen base, with another nitrogen base, right? And frame shift means what? Here we can say addition or deletion of nitrogen base, either one or more than one, right? Now this substitution, because here nitrogen base is replaced, no deletion and no addition is there, right? What happens when it is replaced? That means the sequence is changed. Sequence change means what? Only one amino acid is changed. It is also two types. We can say here two types. First one is called here transition, and another one is called here transversion. Transition and transversion, right? So transition means what? When purine is replaced by pyrimidine, right? Then this one is called transition. And transversion means what? When purine, one thing is there. Transition means what? Purine is replaced by, okay, this is not correct, I found, right? When purine is, purine is replaced by purine, another purine. Look, when both are same, purine is replaced by purine, right? Then it is called transition. And when purine is replaced by or pyrimidine is replaced by purine, then it is called transversion. You understand? Purine is replaced by purine means A is replaced by G or G is replaced by A, right? This side purine is replaced by pyrimidine means A is replaced by either T or C or T or U, right? Then it is called transversion. Second is our frame shift mutation. The frame shift mutation is also two types, as I tell you, addition or deletion is there, right? Addition means here two types. First one is addition addition of one or two. If one or two nitrogen base is added, right, then what happens? The total shift is changed, and this is given in this. We read this addition or deletion. Second one is our deletion, and these are read in our through our NCERT. Ram has read can. You understand what I am saying? Ram has read can. It is given in our NCERT book, and there we will read this frame shift mutation very briefly, right? Mostly, one thing is there. Mostly, this mutation is harmful, right? Okay. So I think we need not to hear once more. Read this one. What is addition? What is deletion? We know if one or two nitrogen base is or nucleotide is deleted, then what happens? The total nitrogen, we can say, genetic code is changed. Just after this addition or deletion point, means all the genetic code, they are changed. That is called frame shift. One or two add, also the total number of genetic codes are changed. They are changed, right? But when it is in deletion or addition occurs in 3 or multiple of 3, then what happens? Only one genetic code is changed. That means only one amino acid either add or delete is there. As for example, sickle cell anemia, right? So that is the example of here. So this is the different type of mutation in organisms. So I think you understand this one. I think you understand this one, right? Now come to the NCERT, right? Because in NCERT we are today, I think, or I want to complete this chapter because only one topic is there, that is DNA fingerprinting. When we consider DNA fingerprinting, in DNA fingerprinting we have 6 steps. First, we need isolation of DNA. Then when we isolate the DNA, then what we should do after isolation of DNA? First, we have to cut down this DNA into fragments. That is called digestion of DNA. When we have different type of DNA, when we get different part of DNA, different segment of DNA, then through gel electrophoresis, right, we get separate the separate size of DNA fragments. Then southern blotting with the help of nylon, right? Then after what happens? That is called then hybridization is there with the artificial probe, and then after we get the X-ray, that is called autoradiography, and through autoradiography we get the DNA fingerprint, right? Now come to the here. This is the last part of our applications and future challenges. Already we did this previous chapter, right? Application and future challenges. Deriving meaningful knowledge from DNA sequence will define research through the coming decades, leading to our understanding of biological system. This enormous task will require the expertise and the creativity of tens of thousands of scientists from varied disciplines in both the public and private sector worldwide. One of the greatest impact of having the human genome sequences may well be enabling a radically new approach to biological research. In the past, researchers studies one or a few genes at a time with the whole genome sequence, and the new high throughput technologies, means when we have a computer database is also there, right? We can approach questions systematically and on a much broader scale. They can study all the genes in a genome at the same time. For example, all the transcripts in a particular tissue or organs or tumor, and how tens of thousands of genes or proteins work together in the interconnected networks to orchestrate the chemistry of life. that is DNA fingerprinting. This is the last one. As stated in the preceding section, that is, we know 99.9% of base sequence among the humans is identical, same, right? Only 0.1, assuming human genome as 3 into 9, 10 power 9 base pair, means in case of haploid, right? Haploid, in how many bases sequence would be their differences? It is these differences, how many it is? It is 3 into 10 power 6, right? It is these differences in the sequence of DNA which make every individual unique in their phenotype appearance. How much is 0.1? It is responsible, right, to make every individual unique in their phenotypic appearance. If one aims to find out the genetic differences between two individuals or among the individuals of a population, sequencing of DNA every time would be a daunting and expensive task. Imagine trying to compare two sets. Two sets means 3 into 10 power 6, right? Base pair. So DNA fingerprinting is a very quick way to compare the DNA sequence of any of the two individuals. This one, 3 into 10 power 6, it is a 0.1%. DNA fingerprinting involves identifying differences in some specific region of DNA sequence, and these specific regions of DNA sequence, they are called as repetitive DNA, which makes the major portion of our DNA. Because in these sequence, a small stretch of DNA is repeated many times, 100 to 1000 times, right? These repetitive DNA are separated from the bulk genome DNA as different peaks during the density gradient through centrifugation. Because density gradient is there, so through centrifugation we can get this. The bulk DNA forms a major peak and the other small peaks. Bulk DNA that is forms a major peak and the other small peaks are referred to as satellite DNA. Small or major peaks, depending on the base composition, either AT rich or GC rich, right? Mostly in prokaryotics, GC rich is there, right? Length of segment, this is the base. What are the base? Base composition, length of segment, number of repetitive units. The satellite DNA is classified into many categories such as microsatellite or mini satellite. This is given here, microsatellite or mini satellites, right? These sequences normally do not code for any proteins. They are not involved in protein coding, means these are non-expressed text. These sequence shows high degree. These sequences normally do not code any protein, but they form a large portion of human genome. These sequences show high degree of polymorphism. Polymorphism means many forms are there. This is the very important for DNA fingerprint. And we can also say here, this is the basic of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, right, skin, bones, saliva, sperm, etc. from an individual show the same degree of polymorphism. Same degree because it is the same animal, same. So they have DNA, same DNA is there. So same polymorphism is there. They become very useful to identification tool in forensic applications. We can get any DNA from any cell of our body. We have same type of mini satellites, same type of microsatellite, right? Further, as the polymorphism are inheritable from parent to offsprings, right? DNA fingerprinting is the basis of paternity testing in case of disputes. Who is the biological? That is here. Sometimes you also see some news in which a dispute is there. Biological, who is the biological father or biological mother is there, right? Sometimes, mostly it is for biological father, for the property and the distribution there. As polymorphism in DNA sequence is the basic of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we have understand what DNA polymorphism means in single term. Polymorphism means many forms. Poly means many and morpho means external structure, right? Variation at genetic level arises due to the mutation, which arise from the due to the mutations, and recall different kind of mutations and their effect that you have already studied in chapter 4. So not chapter 4, just before this DNA fingerprinting, I already tell you how many types of mutations are there, right? This one. And in the preceding section in the chapter, okay, now mutations may arise in an individual either in somatic cell or in germ cell. Somatic cell means it is not inherited, but it never inherited, but it is, if it is present in germ cell, that is right. New mutation may arise in an individual either in somatic cell or in germ cells that generate gametes in sexually reproduction organism. Germ cells, they are called as germinal cells or germ cells. If a germ cell mutation does not seriously impair individual's ability to have offsprings who can transmit the mutation, right, it can separate to the other members of the population. Simply, it can separate, right, through sexual reproduction. Allelic, again recall the definition of allele from the chapter 4. Allele means what? Allele means genes which are located on different homologous chromosome but on the same loci. Loci means same location, right? Sequence variation has traditionally been described as a DNA polymorphism if more than one variant allele at a locus occurs in human population with a frequency greater than 0.01. Right? This is, you have to read this one. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. What is DNA polymorphism? If an inheritable mutation is observed in a population at high frequency, then this one is called as DNA polymorphism. The probability of such variation to be observed in non-coding DNA, right, sequence, that is mini satellite or microsatellite, would be higher as mutation in these sequence may not have any immediate effect because they are non-coding. Non-coding means they cannot code any proteins, right? So no protein means no enzyme. No enzyme means no phenotype appearance is there, right? So they may not have any immediate effect or impact with an individual's reproducibility. These mutations keep on accumulating generation and after generation. They are present in the genes, right? They accumulate. One, another generation means it becomes double, third. As already we read here, in which one? And euploidy, 2n, 3n, 4n, 8n. It is accumulated because they are genes. These genes are accumulated generation after generation and forms one of the basis variability or polymorphism. Variability means variation, new species or polymorphism. There is a variety of different type of polymorphism ranging from the single nucleotide chains to very large scale chains. Single nucleotide chain means that is frame shift, means total protein is changed, right? From evolution, speciation, evolution you know, evolution means what? Gradually differentiation comes. Speciation means forming of a new species. Such polymorphisms play a very important role and you will study these in details in the higher classes. We will read, okay? Evolution also we will listen. The technique of DNA fingerprinting was initially developed by Alec Jeffrey, right, in 1970. He used a satellite DNA as proof that shows very high degree of polymorphism, which he called variable number of tandem repeats, VNTR. The technique as used earlier involved southern blot hybridization using this southern is not a south, right, but it is the name of the scientist. Southern blotting hybridization using radio labeled VNTR as a proof. Radio labeled VNTR means it used phosphorus, which phosphorus P32, right? It includes how many steps are there? Six steps are there. First, isolation of DNA, right? First, we have to isolate the DNA to the cell. For isolation, we use different type of enzymes, right? Then centrifugation, and then we get the DNA, either through centrifugation, or through chilled ethanol. Sometimes we use also chilled ethanol. Digestion of DNA by restriction endonuclease. Then afterward, we digest the DNA. With restriction endonuclease, it cuts the DNA specific region, specific palindromic region. here one thing is there. This isolation of DNA through different enzymes by using of different enzymes except DNAs, right? And through centrifugation or by using chilled ethanol. Digestion of DNA by restriction endonuclease. Okay, here we knew very well because we have ECO, RI, right? And another which is mostly commonly used in human, right? That is HAE3. HAE3 means what? It is Haemophilus AE aegypticus. Three, right? This is a bacteria. Mostly it is used and it cuts the DNA where CCGG sequences, right? So this means we understand and we know restricted endonuclease enzymes, they cut the DNA where palindromic syndrome, that means they are made up of 6 nitrogen base pair or maybe 4 nitrogen base pair, they cut this DNA fragments, right? Mostly we use here HAE3. EcoRI, already we read this EcoRI in our next topic, that is in biotechnology very deeply, right? Okay, now separation of DNA fragment by electrophoresis. So by using electrophoresis means we use gel. Which one? Agarose gel. Agarose gel electrophoresis. Right? Agarose gel electrophoresis. Agarose we know, agarose come from our red algae, that is Gelidium and Gracilaria. So we get agarose, agar, and this agar is a type of sugar. It forms a colloidal solution, right? So through colloidal solution, we know in electrophoresis what happens, because DNA is a negative in its nature, so it moves toward the positive, means toward the anode, right? Positive electrode, that is anode, toward the anode. The smallest fragment of DNA, it moves very fast. It travels long distance. So now through this gel electrophoresis, we get different segments, means different size segments, different length segments are there. You can see here, this one is a these are called well. Well means, well means it is a tank, where tank means very small dips are there. Here we use this DNA, right? DNA are pulled into this one. Then what happens? This side it is negative, this side it is positive. Now what happens? Because DNA are negative, so DNA moves from this side to this side. So the smallest one is that. This side smallest one is there, right? And this side largest one is there, right? So this is the smallest fragment. These are DNA fragments. So we can separate the different length of DNA through gel electrophoresis. Then after transferring blotting of separation of DNA fragment to synthetic membrane, right, such as nitrocellulose or nylon, and this one is called southern blotting. Southern blotting, like an ink blotter, it acts like an ink blotter. For blotting, what we use means near about 1.5 kg pressure on this gel electrophoresis. What happens? These DNA fragments, they are attached with nylon or nitrocellulose membrane, but we cannot see them. We cannot see them because they are very small, right? Then what happens? Then after we have to hybridization using the labeled VNTR. So these are artificial. These one are artificial DNA, RNA proof, RNA or DNA proof, PROV probes, right? Because they have radioactive. Radioactive means they use P32 phosphorus. DNA is negative due to the phosphoric and right phosphoric acid. Okay, when we add this one artificial, because they have radioactive, so when we expose them into X-rays, then what happens? We get different color bodies. Different color bodies, orange, stellar bodies are there. So detection, that is called autoradiography. This detection of hybridized DNA fragment by autoradiography, that is schematic. Okay, representation is given here. Autoradiography means we use X-ray. These are exposed in southern blotting is, or we can say this hybrid DNA is exposed in X-ray. Now we get different bands, right? Different band, we can see this different band, and when we consider the length of different band, then we get the specific view. The VNTR belongs to a class of satellite DNA referred to as mini satellite. That is, a small DNA sequence is arranged randomly in many copies. The copy number varies from chromosome to chromosome in an individual, right? The number of repeat shows very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Kb means what? Kilo base pair. Kilo base pair means what? 1000 base pair. 20,000 base pair. From 0.1 thousand base pair to 20,000 base pair. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands. Already I tell you here, right? Many bands of different size. Right? That is, these bands give characteristics pattern of an individual DNA. It differs from individual to individual in a population, except in case of monozygotic identical twins. Monozygotic identical twins, why it is except here? Why it is an exception? Because we know monozygotic twins, right? They are identical twins because they form from a single zygote, and single zygote means same DNA. Both have same DNA. So these are exceptions, right? They develop from a single zygote, that means from a single type of DNA is also there. So single DNA means we can say identical DNA is present in both, right? The sensitivity of the technique has to be increased by use of polymerase chain reaction, PCR, which we read in the next chapter, right, in biotechnology, PCR. Consequently, DNA, that is also called as amplification, right? DNA from a single cell is enough to perform DNA fingerprinting analysis. We need only, if we have only one micron, yes, we can get, right? In addition to application in forensic science, it has much wider application, such as figure is given. In determining the population and genetic diversities, currently many different probes are used to generate DNA fingerprinting. And here you can see this figure and understand, right? This is the paternal chromosome, this one, and this one is the maternal chromosome. Chromosome number 7 and chromosome number 7, right? Now this is the chromosome 7. You can understand this one belongs, match this one, no. Chromosome 2nd, matching, no matching, right? Chromosome 16, are they matched? We can say no matched, right? DNA from individual A, DNA from individual B. Now this is the DNA from individual A. DNA, that is our, suppose that this one. Number of short tandem repeats, you can see here. This is chromosome 7. Number of short tandem repeats, how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 is given, right? But here what is given? Look here. That C and that, these both are C. We can see. This is C, right? And this AB, we have 2. And these 2, we know, this matched to this. This one matched with this one, right? This 11th chromosome number 7, right? Second, this one, how many is there? 1, 2, 3, 4, 5, 6, 7, 8, 9, and this is also here. And this one is matched with this one. But here, it has no VNTRs. 7, it is present, okay. Here it is A present, B also present. This is present in all these cases, but this chromosome 2 second one is present here, but not present here. This is present here. So when we see this one, what we get? DNA from the crime scene C, right? And amplified, repeated, separated by the size of gene, give DNA fingerprinting. Now you understand who is the criminal. When you match this one and this one, so you can get that which one is matched DNA. This DNA is matched with this one. That means this B1 is the criminal. We can understand. right? And here, representation of DNA fingerprint, few representative chromosomes has been shown in the certain different copy of VNTR. For the sake of understanding, different color scheme has been used to trace the origin, right? The two alleles pattern maternals of the chromosome also contains different copy numbers of VNTR. Mother and father, they are also contains different copy, right? It is clear that the bending pattern of DNA from crime scene matched with the individual B, not with the A. When you see this crime scene, it is matched with the B, not with the A. That means this one is the criminal, right? This one. Now understand? This is all about of molecular basis of genetic inheritance. If you find out any doubt, you can ask. Okay? Thanks.